So, welcome back to Scalpels and Synapses, Demystifying Neurosurgery, a podcast by Mind. I'm Francesca, and I'm here with my co host Samia. Through decades of groundbreaking research, neurosurgical leadership, and mentorship, today's guest has profoundly advanced the field of neurosurgery. Dr. Michael Fehlings is a world renowned Canadian neurosurgeon and global leader in spinal cord injury research, whose work continues to advance recovery and repair in the spinal cord. He has published over 1,600 scientific articles, with his work cited nearly 100,000 times. His work spans clinical and laboratory research as well as global collaboration, ranging from pioneering studies in spinal cord injury and neuroprotection to leading major clinical trials and advancing regenerative approaches, including neural stem cell strategies. Clinically, Dr. Fehlings specializes in complex spine surgery with a particular focus on traumatic and non-traumatic spinal cord injury, as well as spine oncology. He is also a fellow of both the American College of Surgeons and the Royal College of Surgeons of Canada. Dr. Fehlings holds numerous professional appointments, including serving as Professor of Neurosurgery and Vice Chair of Research at the University of Toronto, Co-Director of the University of Toronto Spine Program, and holder of the Robert Campeau Foundation and Dr. Charles H. Tatter Chair in Brain and Spinal Cord Research at the University Health Network in Toronto. He is also a scientist at the MacEwan Centre for Regenerative Medicine and a McLaughlin Scholar in Molecular Medicine. His work has been recognized through prestigious honors including the King Charles III Coronation Medal, the Reeve Irvin Medal in Spinal Cord Injury, and the Olivier Krona Award, the top award internationally for neurosurgeons and neuroscientists awarded by the Nobel Institute at the Karolinska Institute in Stockholm. In this episode, we'll explore Dr. Fehlings' journey into neurosurgery, dive into his research on spinal cord injury, and hear his insights on the future of the field, along with advice for students interested in pursuing neurosurgery. Dr. Fehlings, thank you so much for joining us today. I'll let Samia take the lead.
Thank you, Francesca. So to start off, we would love to talk about your journey into neurosurgery. So you completed an MD and neurosurgery residency at the University of Toronto. So what first drew you to pursue neurosurgery?
I would say that I was seeking a challenge in life, and I wanted to pursue an area that I would find challenging in a number of ways. I knew that I wanted to pursue a surgical area, and because the practicality of doing surgery appealed to me, and I was drawn, to the brain and the nervous system, and I found that the complexity had a lot of appeal.
And did you make this decision early in your training, like early in your medical journey, or was it like so much later on?
Well, I was attracted to neurosurgery very early on. I did a lot of electives in the first couple of years of medical school to kind of explore a number of areas, mainly electives in different surgical disciplines. And I liked a lot of different areas in medicine. And the first 2 years of our medical school were kind of more in class learning, and then years 3 and 4 were more practical. And when I was exposed to clinical neuroscience, neurology, neurosurgery in my third year, that really kind of galvanized my interest in pursuing neurosurgery.
Amazing. And to inform like all the people who are currently listening, during your residency, you also completed a PhD, becoming one of the early surgeon scientist trainee at the University of Toronto to do so. And your work focused on the pathophysiology and treatment of spinal cord injury under the supervision of Dr. Charles Tater, who is also a pioneer in the field. Following your residency, you went on to complete a postdoctoral research fellowship at NYU Medical Center under the famous Dr. Wise Young, and then a clinical spine fellowship under Dr. Paul Cooper. And again, my next question is, what drew you specifically into spine as a focus of your career?
Yeah, that's a great question because I was really attracted to neurosurgery because of the brain and the central nervous system. And of course, I was aware that the spinal cord is an important component of the central nervous system, but I wouldn't say that when I started neurosurgery that I had a particular specialized interest in spine and spinal cord injury. And this was really, I think, being in the right place at the right time, if you will. So my first rotation as a neurosurgery resident was working under Dr. Charles Tatter, who had a big interest in spinal cord injury. And I was really struck by the devastating aspect that spinal cord injury would have on people who were usually in very good health and then would have an injury, would have perfect cognitive functioning, and then were profoundly impaired. And I wasn't particularly thrilled with the lack of treatments that were being offered at the time. And sometimes, people need to make a choice. You either avoid that area and say, well, you know what, I don't really like this area, or you can make a decision to try to do something about it. And Dr. Tatter was running a basic science laboratory, and he was very interested in looking at the pathomechanisms of spinal cord injury. And this really drew me in. And I thought that to try, if one was going to make a difference in the treatment of patients with spinal cord injury, that further research would be required. And I had the opportunity to do basic science research in Dr. Tatter's laboratory during my residency. And then I made the decision that I was quite interested in the area and that I was going to pursue a PhD, which was quite challenging to do during residency, but nonetheless, that evolved. And so Dr. Tatter had a big influence on me. And the other individual who was strongly influential in my career development was the chairman of neurosurgery at the time, Dr. Alan Hudson. And as I was emerging out of the PhD, Professor Hudson said to me, look, the whole area of spine surgery is evolving, and, would you be interested in focusing in this area? And I thought about that, and then decided that made sense, that I would, pivot, if you will, and try to focus in the area of spine surgery, and spinal cord injury. And so that was really, partly driven by, the patients that I was seeing, and then also by key mentors.
We can see that finding good mentors is really important, especially when we have to choose or orient our career in like such a competitive specialty. And building on that, we want to ask you, like, for students who are currently looking for mentors in the field, what advice would you give them on finding the right mentorship and to build those relationships that you described early on?
Yeah, so I think there's a growing recognition of the importance of mentorship, and I think mentorship has been kind of a key longstanding principle in the education of doctors and scientists. But there are more formal processes now, and so there are formal mentorship programs that universities will offer, and that the medical school and residency programs will offer. So I would say, firstly, take advantage of these. Um, but I often find that finding the right mentor and having the right mentor-mentee relationship is also, there's an element that's kind of an organic process. So sometimes you're drawn to people, you're drawn to their personality, you like the area of work that they're doing, and I would encourage people, to be open to this type of an experience. And then in addition, sometimes, you can be drawn to someone. That doesn't necessarily always mean that you're going to be, their exact mirror image and that you're going to be doing exactly what they were, interested in doing. So in my own experience, I've mentored many people, and many of whom have ended up pursuing spinal cord injury research and spine surgery as a subspecialty, but not all have. Others have gone into other areas and will, kind of draw on certain elements that you're a proponent of. So, it's kind of a mixture then. There's formal processes, but then there's sort of the organic process of linking with someone that you're kind of drawn to.
Thank you so much for your answer, Dr. Fehlings. I'm just gonna, no, sorry, I'm a French speaker, so sometimes I do mix English and French. Now I'm gonna let Francesca take over the next section. Thank you.
Thank you. So our next section is titled Bench to Bedside. So we'll be exploring your research journey from fundamental laboratory discoveries through clinical translation and its impact on advancing human therapies and informing updated clinical guidelines. So we read a bunch of your papers and we're obviously big fans. And so one of your papers we wanted to discuss was the 1996 Journal of Neuroscience paper, where you explored mechanisms of secondary injury in spinal cord axons, particularly the role of sodium in driving ongoing damage after the initial trauma. And in that study, you showed that in rat models, an increase in intracellular sodium plays a key role in worsening the injury, and that blocking sodium entry can actually can improve recovery of axonal function. And this work was especially important because not only did it help shift the understanding from the more common understanding that it was predominantly calcium driving the damage in secondary mechanism, but also it ultimately laid the groundwork for the development of major international multi-institutional clinical trials, notably the riluzole spinal cord injury study and the efficacy of riluzole in surgical treatment for cervical spondylotic myelopathy trial. So my question is, for trainees and aspiring surgeon scientists watching this, we're really curious, how do you begin identifying a meaningful research question to study in the lab, especially so early on in your career?
Yeah, so that 1996 paper was quite an important paper in my career. It was really the first kind of major paper coming out of my independent basic science laboratory. And it has paved the way for subsequent translational work and ultimately clinical trials, which I can kind of briefly discuss. But, what was the path toward that? And I was really drawn to a fundamental question, and that was when someone has an injury to the central nervous system, either a brain injury or a spinal cord injury, there was sort of the sense that when I was early in training, that's it. You have the injury and you can't do anything about that. And I kind of challenged that notion. And in my training with Dr. Tatter in the laboratory, we were pursuing a concept which is now referred to as a secondary injury. And the idea that there's a primary mechanical injury or some kind of an injury to the brain or the spinal cord, and that the impact of that injury is amplified through molecular and cellular and vascular processes that occur after that. And we were particularly interested in the idea that after spinal cord injury, there's a reduction in blood flow to the injured site, and that ischemic damage then would then trigger secondary molecular and cellular events. And so that was and we in fact pursued that, and the secondary injury hypothesis, as it was then referred to, has been, I think, very well accepted now and has been validated a number of ways. And so the question around the sodium and the calcium evolved really from that initial research and the discoveries around the role of ischemia. And so I asked the question, okay, well, what happens when tissues are ischemic and the cell membranes are damaged and typically cells swell? Well, why do cells swell? Well, sodium goes into the cell and water goes with the sodium because of the sodium hydrogen exchanger. And so it was really a very simple question. And during my postdoctoral fellowship at NYU, I acquired skills to dissect spinal cord tissue from rat models and mouse models, and to keep it um, uh, alive in a dish. And then I could study this at a very detailed level, electrophysiologically and molecularly, and it was very easy to alter the concentrations of salts and ions and so on in the extracellular media. And we then explored this question, okay, why are the cells damaged in a secondary fashion? And it turns out that, for sure, sodium was a key event, and that it subsequently triggered a series of secondary events. And so, sometimes, the questions that evolve arise from, a clinical issue, and then asking the question and then going back and thinking, what are the fundamental mechanisms that might be underlying this, and then having an understanding of this. And then that discovery that sodium was critical to the secondary injury process then led to efforts to try to mitigate the injurious effects of sodium entry. And then that subsequently led to clinical trials with a drug called riluzole.
That's great. Thank you so much for your insights. And another question relating to this, once you have a promising discovery in the lab, how do you then go about translating it into a clinical trial with humans with multi-institutional collaboration? If you could walk us through like the steps involved in that.
Yeah, so that's it's a process that is fairly involved, and one needs to be prepared to be persistent. And I think that persistence, is important. And I think kind of one of the take-home messages that I would have is that there's lots of ups and downs in life and in your career, and just to recognize this, but to kind of have your eye on a particular set of goals. So were several elements that were important to translating this fundamental discovery that that sodium entry was critical to the secondary injury process. So the first step was to try to develop a pragmatic approach to block sodium entry in a way that would be safe and effective. And so this was then done with in vivo animal models of spinal cord injury. So one needs to have a model of injury to validate that model, and then we needed a drug. And the drug that we ultimately selected, which is now referred to as riluzole, was particularly attractive because it targeted a particular subtype of sodium channel called a non-inactivating sodium channel, and the drug itself was very safe. So it did not have any cardiotoxic or neurotoxic or other side effects. And so we then validated this in animal models. This was then replicated by other labs. And so we had a strong basic science discovery. This was then translated into animal models that in many ways mimicked what was occurring in human patients. And then you need to convince colleagues that this is an attractive strategy, and that involves the creation of clinical trials networks. And I benefited from um, uh, um, two clinical trials networks that were established relatively early on in my career. One was a spine trauma study group. The main target there was to focus on the role of early surgical intervention to attenuate secondary injury. And the second study group that emerged from that is still quite active is the North American Clinical Trials Network. And through NACTN, or the North American Clinical Trials Network, we then engaged colleagues in another clinical trials network that was emerging through AO Spine, which is a large international NGO. And collectively, we went through quite a rigorous process to evaluate emerging translational treatments for spinal cord injury. And after a rigorous process, it was felt that riluzole was attractive to move forward. And then we went through a process to do early phase clinical trials to confirm the safety of the drug and to get a sense of potential signal, and then moving into what are referred to as phase 3 randomized trials. And that's a fairly involved process, and it involves writing grants and getting consensus and then moving forward with the grants. But that's you know that's a short and sweet description of a fairly detailed process to try to move that forward. But it all started with a pretty simple question, right? Is a primary injury amplified through secondary injury mechanisms? Yes, it is. Looks like there's an ischemic injury. How does the ischemia cause cell injury? And then turns out that sodium is a key player. And, so sometimes, the best questions to ask are ones that are quite simple and fundamental.
Thank you so much. It's really, it's great. It's a lot. It sounds like a very detailed process to go through, but it's definitely worth it to advance the field. And building on your earlier work on secondary injury, you went on to lead the surgical timing in acute spinal cord injury study, which you briefly touched on. So in that study, you looked at patients with traumatic cervical spinal cord injury and compared those who underwent decompressive surgery within 24 hours to those who had surgery later, and then followed their neurological recovery over time. And it's really interesting how this connects back to the biology of that earlier study in 1996, where now we know that there is this secondary injury cascade after the initial injury. And the idea is that by decompressing the spinal cord earlier, you may be limiting that secondary injury process. And so your study did show that patients who had earlier surgery were significantly more likely to experience meaningful neurological improvement without an increase in complications. And this work contributed to the growing body of evidence supporting early decompression after spinal cord injury. So my question is, once you had those really great clinical results after this long multi-institutional study, how do you then go about sort of convincing your colleagues that this is the new guideline, like this should be the standard?
Yeah, so that's been an interesting process, and to be honest with you, it's an ongoing process still. So when I was a resident in training, and at the outset of our discussion this morning, I indicated that I was exposed to people with spinal cord injuries and spine trauma, and at that time, the patients were largely managed non-operatively. They were on bed rest, they were put into traction, usually put into a brace or a halo vest, and surgery was done, but not very commonly. And I wouldn't say that the surgical techniques that were available were always satisfactory. And there was during the course of my training later on, and then early in fellowship, and then early in my experience as a staff person, there was really a revolution that occurred in advances in spine surgical techniques and neuroanesthesia and critical care and so on. And it allowed us to be able to operate on patients early and safely and to establish stability. And I always felt that the secondary injury process was important. But the idea of actually operating on people with spinal cord injuries and doing this early and doing the decompression, this kind of went against the standard thinking at the time. And so there was a fair bit of resistance that was involved. And so I referred earlier on to the Spine Trauma Study Group. The Spine Trauma Study Group emerged through a collaboration between myself and a number of other individuals, most prominently Alex Vaccaro, an orthopedic spine surgeon in the US. And we both shared a passion for the idea of early surgical intervention in patients with spinal cord injury. And we created the Spine Trauma Study Group. And the STASCUS study, which emerged, was really the cornerstone of that study group. And we examined the hypothesis that early surgical intervention was safe and could improve the outcomes. And then we showed, sure enough, that there was a very large effect size that we saw. And the STASCUS trial changed management around the world. But there was pushback. Some people felt that, well, I'm not sure that we're going to be accepting those results. But what occurred after STASCUS was that there was an explosion of interest in the whole topic of surgery and surgical timing. And then subsequent to that, we developed an initial set of guidelines that was published in 2017 by AO Spine and the Cervical Spine Research Society. And there we showed for the first time that, yes, early surgical intervention is the preferred management strategy. But we also identified a number of knowledge gaps, and we felt that although the evidence was pointing this direction, we needed even better evidence to do that. And then this subsequently inspired a number of us, including my own group, to undertake some higher quality studies, one of which was published in the Lancet Neurology, studying over 1,000 patients with spinal cord injuries. And then a couple of years ago in 2024, we published a revised set of guidelines for spinal cord injury. Now the evidence was allowing for a strong recommendation for early surgical intervention. But we also identified potential barriers and further knowledge gaps. And so there's a process called the knowledge to action cycle, which is it's like a cycle. You start with a question, then you develop some evidence, and then you try to develop consensus. And if there's enough evidence, you can develop a guideline, but then you need to validate that guideline. You need to test drive this, and you're constantly evaluating this. And there's a big process involved with knowledge dissemination and knowledge translation, right? And it's this kind of an iterative process over time that's required to try to change the care pathways for patients with any kind of a condition in medicine. And this was the path that we have followed and continue to follow related to the role and timing of surgical intervention for spinal cord injury.
That's great. It's really interesting. It's kind of like a domino effect where one really major study sort of inspires so many other people to study the same topic, and then that could eventually change the practice. So my last topic in this section would be about your work in stem cell research. In 2006, you published a paper in Journal of Neuroscience, and the study explored the effects of transplanting neural precursor cells from mice into the injured spinal cords of rats to see whether they could promote repair. And the study found that these cells were able to survive, integrate into the spinal cord, and differentiate into oligodendrocytes, cells responsible for producing myelin. And importantly, they were able to remyelinate the damaged axons and were associated with improved functional recovery. And so more recently, you and your colleagues published a 2025 Nature Neuroscience study in a mouse model of spinal cord injury demonstrating that enhancing AMPA receptor signaling can increase the activation and migration of endogenous spinal cord stem cells and lead to improved functional recovery. So the field is clearly it's continuing to evolve, and there's so many great results that are coming out from you and your group about stem cell research. And since the area is still developing, how close would you say we are to actually seeing stem cell based regenerative therapies become a part of clinical practice in spine? And how might they change the way we view the prognosis of spinal cord injury or treat it? And what are the key barriers that we still need to overcome?
Yeah, thank you. You've done your homework. So, yeah. So, taking a step back, I got interested in stem cells um, around a very basic question initially. And one of the discoveries that I made in the lab was that even after a severe injury, there's often some residual neural tissue that's present at the level of the injury. And it turned out that if you could preserve around 10% of the axons at the site of a spinal cord injury, this could be associated with substantial restoration of function. And one of the challenges was that the axons, although intact, were often demyelinated. And this demyelination contributed to the loss of function. And so I got drawn to stem cells, specifically neural stem cells, based on a few key discoveries that have been made. And one was that there actually are neural stem cells in the adult central nervous system. So, there's this dogma that the adult mammalian CNS, human brain, spinal cord, don't regenerate. And that's largely true, but not entirely true. There is some limited regeneration that can occur. And there are actually stem cells in some areas of the brain, and particularly in the subependymal zone of the subventricular region. And there was a key discovery made by Weiss and Reynolds in Calgary around the fact that these neural stem cells exist. And then there were protocols developed to isolate these neural stem cells. And then I thought, this could be an interesting question to examine whether if we use these neural stem cells, could we remyelinate these axons? And this was the genesis of the 2006 Journal of Neuroscience paper, which was the first kind of big stem cell paper out of my laboratory. And this has now driven a significant translational effort that's moved forward. And we've even had initial phase clinical trials with neural stem cells, and we've learned a lot from this. We published our work in an article in the Lancet Neurology. So, this is a process where we kind of went from the bench and we went to the bedside. We did clinical trials. We found that the neural stem cells were safe and they were well accepted. We didn't see complications, and the effect sizes were encouraging, but they were too small to move forward. And we felt that we needed to go back to the bench. And this is what we've now been doing. And we've been studying neural stem cells quite intensively in the laboratory. And there have been some really major discoveries that have occurred in the last 15 to 20 years. One is that we don't really need to use embryonic sources of stem cells anymore. So that's sort of not the current technology. We can actually use somatic cells, so, for example, a skin fibroblast or a blood cell. And you can reprogram these to create a neural stem cell. And Yamanaka in Japan received the Nobel Prize for this. It's called induced pluripotent stem cells. So that's a big discovery, right? So one is you can avoid all the ethical issues, et cetera. But the other side of this as well is that potentially you could use a person's own tissues to create a stem cell that's unique for them. So that's very cool. And then the next discoveries that have been made are that one can actually use these neural stem cells as a gene therapy platform. And you can actually alter the neural stem cells in a very safe way to deliver molecules. So right now we're studying the effects of a growth factor called glial-derived neurotrophic factor. And we're also studying the effects of a particular enzyme which degrades the scar tissue around the injury site. And so now we're trying to gear up to the next round of clinical trials, working very, very hard to try to move this forward. And I'm hopeful that we'll be getting into the next round of early phase clinical trials in the next 2-3 years, to try to move this forward. And so, this is kind of a longer term strategy, but the idea here, kind of looking at methods to try to actually regenerate, lost central nervous system tissue. And this has huge implications for people with spinal cord injuries, brain injuries, and all sorts of other conditions, including neurodegenerative uh, conditions, ALS and Alzheimer's disease, and so on. Now, the paper that you referred to that we published last year, the first author was actually a neurosurgery resident, Lorraine Hatcham. That was her PhD thesis. And that work really emerged from the idea that there actually are neural stem cells in the central nervous system. And we had hypothesized that there were neural stem cells in the center of the spinal cord, in the central canal. And there were other lines of work from our group, from others, including Jonas Friesen, the Karolinska, that gave some background that suggested that that might be, might be in fact the case. And then we kind of unlocked some of the molecular pathways that are involved in triggering the proliferation of these neural stem cells. And so that's really quite interesting work, because it suggests going forward that we might actually be able to use a person's own endogenous neural stem cells to repair and regenerate the injured nervous system. And so that's an area of work that is continuing to be pursued.
It's really exciting work, and I'm really looking forward to see the outcome of all your work in stem cell research. So that concludes my section. I'll let Samia take over for the next one.
Awesome, thank you. So our next section is called Lessons from a Surgeon Scientist. Basically, we will focus on insights and reflections from your career. So you have published extensively in leading journals, including work in Nature, as even Francesca mentioned. Do you have any advice for students and trainees who are hoping to contribute to meaningful research and also to publish in high-impact journals?
Yeah, so I would say that research training, I think, is really integral to the training of a surgeon. And I think research is very integral to the training of the neurosurgeon. And neurosurgery really has its origins in clinician scientists such as Wilder Penfield in Montreal, right? Who kind of examined basic questions and then tried to translate these into treatments. And so there are opportunities as a medical student to pursue research electives and research training. So I would encourage medical students to take advantage of this opportunity. And many residency programs in neurosurgery do offer the opportunity to embed research training in that residency program. So at the University of Toronto, we have a clinician scientist training program, which includes actually all the surgical disciplines, including neurosurgery. And so residents are given the opportunity to, similar to my own path, to do a thesis-based graduate degree during the course of their residency. And so this, potentially opens the door for trainees to, to try to, pursue, an area of research training. Now, not all neurosurgeons are going to be doing research. Not all are going to be at universities or pursuing an academic course, and that's fine. I mean, there's lots of different career paths that people can pursue, but I still feel that it is important to have research training as part of the training because it allows you to be open to new knowledge that is coming forward. And, the practice of medicine in general, and certainly neurosurgery, is evolving very quickly. And so, in order to embrace those changes that are occurring, it's important to have knowledge of the scientific processes and be able to evaluate the literature and to evaluate and incorporate new knowledge as it's emerging. So, I'm quite a proponent of the value of research training in the training of physicians in general and certainly neurosurgeons.
Thank you so much for your thoughtful answer. And just to follow your answer, we know that throughout your career, you have combined like a busy clinical practice, and also you were able to lead a highly impactful research program. How do you balance clinical work with research?
Yeah, so life is all about balance, right? And you know, the notion of balancing work and life is important, but I would say that it's really about life balance and not necessarily work-life balance, because the concept of work-life balance probably doesn't apply to a neurosurgeon, because one of the decisions you need to make when you become a neurosurgeon is that, your profession will be part of your life, and it's a passion. And so I derive personally a lot of enjoyment and reward out of different elements of my professional life. And so I've made a lot of friendships, um I find doing research to be quite a creative process, right? And so I think one of the points is to understand that different professions in medicine will require a different level of commitment, and neurosurgery requires a high level of commitment. So that's the first point to understand. Now, the I've had a family. We have 3 kids. We have grandkids. It definitely is possible to have a family and to, enjoy your family. And it's very important also, I think, to take time off for your family and for your friends, and importantly for yourself, right? So I think that's part of this. And I think that's um, probably the fundamental question of what to do. And then in terms of your professional life, how do you balance doing research in clinical work? I think it's a question then of, making certain decisions in terms of how you schedule your life and schedule, your week. And so typically in my week, I have certain days that are dedicated to clinic. Usually it's one day a week of clinic. It's quite a busy day. And then I'm in the operating room 1 to 2 days a week. And so this on average leaves about 50% of my week for, academic work and for research work, and about 50% of the time for clinical work. The reality is that, there's no such thing as being a 50% clinician. So you're always there for your patients and so on. But it's a matter of kind of how you schedule, kind of certain, kind of areas of activity. And then it's also important to make sure that you balance this with, you're allowed to take vacation. You're allowed to, take time for yourself, and you need to do this. And then also, if, to balance things out for your friends, your family, and so on. But for me, the concept here is really about life balance, right? Because being a neurosurgeon, it's different. And so, sometimes, I will I enjoy reading academic articles, I enjoy writing and so on. And so, for me, this is also kind of a creative type of an activity. And so it's kind of a matter of how you kind of look at this kind of yourself.
I really like this philosophy of thinking. I feel like once you decide something that you really like, you're not gonna feel like it's work. Work is not work anymore. So that's what I'm understanding from your answer. Thank you so much for answering my questions. It was all for this section on my behalf.
Very good.
So we're gonna start to wrap up, but we have just one last question. So Dr. Fehlings, you are truly a pioneer in this field, and your work has inspired so many, including us. Our last question would be, what advice would you give to medical students like us who are interested in pursuing a career in neurosurgery?
I would say pursue your dreams and, you know, uh, be aware of what you're doing. And I say this in a positive sense, but it does require a commitment. And, at the outset, you're asking what drew me. I I was drawn to neurosurgery because I thought it was really cool, but also I was aware that it was very difficult and I really wanted to challenge myself. And so I think that, everyone is different. Um, but I would say that, neurosurgery is incredibly rewarding. And I think it's the right fit has to be there. And so you have to really assess in yourself whether, you're not just drawn to this conceptually, but that you're also, prepared to make the personal commitment, both in terms of the training that's required and then also the personal commitment. If you're, open to doing that, then it's an incredibly, rewarding experience. And, one of the things that was drawn to me is that, you're taking care of patients that are very often in an incredible crisis. And you have the opportunity using your skills and your knowledge to make a huge difference in the lives of these individuals. And that's an incredibly rewarding experience. And so I think that it's all kind of a matter, it's all a matter of fit. And I think it's not so much what other people will say. I have people say, "Oh my God, how can you work so hard? How can you do this? How can you do that?" But I don't perceive it that way. For me, it's the right fit for me. I really enjoy doing what I'm doing, right? And it's, as I said, it's about life balance, and it's about embracing this profession as part of, your life. And so I think you have to kind of, to look at this. And um, a few years ago, I had the privilege of being the Penfield visiting professor at McGill University, and one of the gifts they gave me was a wonderful autobiography by Wilder Penfield, a famous Canadian neurosurgeon, called No Man Alone. And he described his own path, and he would go for long walks in the woods, and he would try to figure out, the answer to a difficult a problem. And that really resonated with me because I have done a similar thing. So when I was trying to figure out, okay, am I going to be a neurosurgeon? I actually went for a long walk and was trying to figure this out. And when I was faced with the dilemma of these patients with spinal cord injury and, what was I going to do? Again, a similar kind of path. And so I think it's a matter of being open to these different life experiences, and then being honest with yourself and reflecting and taking the time to reflect, and then trying to figure out if the fit is there. And so it's a different fit for every person, but those are some personal reflections that I would share.
Thank you so much. It's so amazing. Every answer you've given has really been incredible, and this conversation was really amazing. I'm sure the viewers will really appreciate it as well. Thank you so much for taking the time as well. We know how busy you are. We were really excited to see when you were willing to participate. But yeah, thank you again. We're really honored to
Okay,
have you as a guest.
okay, my pleasure. Take care. Thank you.
Thank you so much.
Thank you so
Thank
much.
you. Thank you.
Bye.
Bye-bye. Bye-bye. Bye-bye.